Hello, my name is Luis Gomez, and I'm a postdoctoral associate at the Duke Medical School. And so, first of all, I would like to thank you for your interest in this talk. And today I'm going to talk about computational techniques that I'm developing to improve the targeting and precision of transcranial magnetic stimulation, or TMS, a non-invasive brain stimulation technique. Specifically, I hope that you can learn about TMS and get an illustration of how computational techniques enable the development and optimal deployment of medical technologies. And so, first of all, what is TMS? TMS is a non-invasive technique where one or more coils driven by low-frequency current pulses placed on the scalp is used to generate magnetic fields, which in turn induce electric fields in the conductive brain matter. These electric fields then in turn modulate brain activity. Specifically, brain regions with large electric fields are typically stimulated by TMS. TMS is used in cognitive neuroscience to study brain function. So for example, a neuroscientist might use the coil to stimulate a cortical region that is thought to be responsible for finger movement, and then they'll watch for a motor response. It is also used as an FDA-approved treatment of depression and for pre-surgical mapping. So there are many other TMS-based therapies being researched. And so one limitation of transcranial magnetic stimulation is that TMS coils fuse in shallow regions of the brain, and this limits the resolution of functional brain regions that can be resolved by neuroscience experiments and surgical brain mapping. Furthermore, gaining better control of TMS fields could result in more effective TMS therapies. So here I show an illustration of the stimulated region during TMS on the left side. And so we can see that the stimulated region is very wide or has a large spread, and it's very shallow or has a very small penetration depth. And so what we would actually like to see during TMS, it's what in electromagnetics we would call a pencil beam type pattern of stimulation, where you have a very small spread and a large penetration depth. And so the stimulated region looks like a pencil beam. And so more precisely, what we want to do is to increase the precision of targeting of TMS. And this, of course, to enable the development of better TMS therapies and increase the resolution of neuroscience experiments that use TMS. So another limitation of transcranial magnetic stimulation is that there's a lot of variability in between TMS sessions, and this results in variability in the TMS outcomes. And so the variability in the TMS outcomes could be caused by coil placement error and suboptimal placement of the coil on the scalp. So currently in a clinical setting, a cap is placed on the head, and the cap has markers where the coils should reside. These markings might not be directly on their targeted neural sites, or there can be operator error, which causes variation in the TMS-induced electric fields. There are more advanced neuronavigated TMS systems that use MRIs along with cameras and robotic arms to virtually remove the error in coil placement. However, these are expensive. And more importantly, we do not know to what extent coil placement uncertainty results in variability in the electric field induced on targeted cortical sites. And so if it turns out that the small coil movements do not result in a large variation in the electric field induced in the targeted cortical regions, then it might not be so important to control for this coil movement. And these neuronavigated systems might not be necessary. If it is the case that the fields are very sensitive to coil positioning, then neuronavigative procedures would be absolutely essential. And so now that you know what TMS is all about, I'm going to tell you about my contributions and specifically how my work deals to address these limitations. And so in a nutshell, I developed a procedure for synthesizing new TMS coils that permit the generation of electric fields that achieve fundamental trade-offs between focality, depth, and energy. And then I use this to design coils which are twice more focal than existing ones. This is the first improvement in focality of TMS coils in decades. In addition, I developed an uncertainty quantification method designed specifically for TMS that informs us about the sensitivity of TMS procedures to various parameters such as coil position and patient-to-patient variability. And then I used this framework to assess the need for these new MRI neuro-navigated TMS systems. Finally, we are developing computational tools to determine optimal coil placements for targeting fields in the brain. I'm going to briefly talk about all these, starting with the coil designs. And so here I show a picture of existing coils, and these coils have been ranked in terms of their spread and depth of stimulation. Smaller spread means more focal coils, and we would like to have a small spread, so we want to be as close as possible to the horizontal axis on this graph. And as we move in the horizontal axis, we are increasing the depth of stimulation. And what we can see here is that there are a number of coils that sit on a curve that sits closest to the horizontal axis, and they are currently what's considered the state of the art, and they mostly consist of what's commercially available or figure-8 coils. And so to improve on these figure-8 coils, what we do is we use closed-loop optimizers to determine the optimal winding patterns. Then we 3D print a 3D print deformers with groups where the coil windings will reside. We wind our coil, and as it turns out, a typical result is that our coils are more focal than standard figure-8 coils. And so the way our optimization works is that we choose a coil support. So this can be any shape. So the coil windings can sit on a flat, spherical, a hemispherical, or any kind of shape that you can define as a triangle mesh, and a head model. And then we specify a head model. This could be MRI derived or a generic spherical head model. Then we choose a targeted location that the coil must stimulate, a stimulation threshold, or the total electric field that's necessary for a region to be labeled as stimulated. We specify a maximum electric field that the coil can generate in the brain, and maximum energy that the coil can use. Then our algorithm finds a coil which minimizes the stimulated volume while achieving those objectives. And so here I show a drawing of a coil that's representative of a typical design coming out of our algorithm. And more importantly, when we design our coils designed by our algorithm, roughly half the spread for any penetration depth. And again, I repeat, this is the first significant improvement in focality seen in decades. These results here were attained assuming that we can use arbitrarily high energy. However, practical designs have an energy budget because the amount of energy delivered to the brain is small relative to the current flowing in the windings. So we are limited by the amount of current that can physically flow through the wire. And as it turns out, as the energy is decreased, we lose some performance relative to our best designs. And this red curve that I show here starts to approach that of the figure-8 coil. And so here, for a fixed depth, I look at how the spread varies with the amount of energy that these coils can use. Each curve corresponds to a different coil support type. So the black would represent a coil whose windings sit on a flat or a square support, the green for a half sphere support, and the blue for a spherical support. And we can see for all of these curves, as the energy increases, the spread approaches the value of the energy on constrained results. Furthermore, there's higher focality for the same energy and depth of a figure-8 coil for any of these designs. And to achieve the same focality as a figure-8 coil, we require less energy. In fact, there's a reduction of up to 10 times in energy. And so what we see here is that the spherical or morphoconformal coils are more energy efficient or have better trade-offs with spread and energy than the flat ones. However, when it comes to a experimental setting, placing a spherical coil on the head and placing it on different targets, it's quite difficult. And so this is not really a feasible solution. And so to deal with this, we came up with a hat-like shape. This shape was derived by using a number of subject MRIs and finding the maximal conformal shape that enabled placement anywhere along the motor strip. And here, its spread and versus energy is better than that of the flat coils and slightly worse than that of the spherical coils. And so this hat coil shape enables us to maintain good energy trade-offs while achieving improved focality. So once we had our design, we built one to match the depth of a standard figure-8 coil, commercial figure-8 coil. And the way we built it is we 3D printed a former with grooves where the windings reside, and then we wound 2 layers of Litz wire and measured the electric fields using an electric field probe we developed in-house. And the measured results match what we expected from our predictions. More importantly, the electric fields are more focal than those of figure-8 coils. These coils are approximately 30% more focal. And so we can see here that the electric fields measured and predicted are along the vertical axis, and the hat coil versus the figure-8 coil are along the horizontal axis. And we can see that the hat coils generate much more focal field than the maximum coils, and that's seen by the smaller spot size in the electric field maps. And so what distinguished our approach from previous approaches is that it is nearly fully computational. The design was done by an optimization algorithm. The manufacturing was mostly done by a 3D printer. By using computational approaches, we came up with winding patterns that were optimal, but not necessarily intuitive. And then 3D printing technologies enabled the manufacture of more complicated shapes. This, in the end, resulted in coils that stimulate 2 times less volume than conventional coils while matching the same targeted depth. So these computational approaches for design enabled the optimal deployment of TMS. Eventually, this technique could be applied to develop personalized coils that exploit the local gyral and sulcal geometry of the cortex to deliver more focal and more targeted stimulation. So this concludes the design part of our talk. Now I'm going to talk a little bit about the uncertainty quantification. And so what we wanted to do here is, given a probabilistic description of the TMS setup, the uncertainty in the coil placement, uncertainty in the tissue conductivities, uncertainty in the size of the individual tissues, and others. And given all of this, we would like to generate probabilistic predictions for the electric field. Specifically, we want to determine the mean, standard deviation, and other statistical parameters of the electric field. Then we would like to use this framework to determine parameters that the result is the most variability in the electric field. And so, in principle, doing this UQ is quite straightforward using what's called a Monte Carlo method. And so, for example, let's assume that we have 2 uncertain parameters, the coil position along the X and Y direction. And so the coil can be centered about any point on this green square that I show here. So the way that we could uncertainty quantification is that so we would center the coil at a bunch of different positions. And then for each of these positions, we can run a simulation to come up with the electric field that's generated at a targeted cortical site. And then once we have this ensemble of electric field simulation results, we can just compute statistics on them to derive probability density functions, cumulative probability density functions, and then we can extract means, standard deviations, and other statistical parameters. Now, a problem with this approach is that it requires many simulation, and as it turns out, to go from coil position to electric field, it takes roughly 5 minutes for a single simulation. So a quick aside, I've made this repository, and here's the website, and it has all the methods currently used and some new ones for analyzing fields generated during TMX. Feel free to download. And so again, running this Monte Carlo requires many simulations, and because of this becomes computationally intractable because we cannot run a high number of simulations. And so to avoid the high cost of Monte Carlo methods, what we do is instead of using our electromagnetic solver, we construct a polynomial surrogate that we can evaluate rapidly. So we first choose a few points on the random domain. And for these points, we run a simulation. So these are very few points, so we can run a simulation for each of these. And then we construct a polynomial fit around these points. And then once we have this polynomial fit, we run the Monte Carlo on the surrogate or the polynomial fit. And this enables us to get the statistics quite rapidly. And so whenever there's a rapidly varied function, what we use is what's called a multi-element probabilistic approach, which adaptively subdivides the random domain and introduces local polynomials in order to be able to capture these rapid variations while minimizing the number of simulator evaluations that are required. Furthermore, for increasing number of random variables, we use what's called a high-dimensional model representation techniques, which introduces decoupling between variables iteratively, thereby minimizing the total number of simulations required to achieve an accurate polynomial for high-dimensional problems. Okay, so now that you know all about my method, I'm going to show you some experimental results. And so as a first study, we consider coil placement uncertainty. And so the center of the coil for different realizations can sit anywhere on this green circular patch. And the radius of this green patch depends on the type of procedure that is being used. So for clinical procedures using this cap and place method, the uncertainty, the radius of this would be between 10 to 15 millimeters. And for neuronavigated ones, it would be between 2 to 5 millimeters. And so here we consider the average electric field on an ROI as our observable. And what we observe, and here I show the average electric field for different levels of uncertainty in the coil position and the 90% confidence interval. What we observe is that the clinical procedures have a variation of about 30 volts per meter, whereas the neuronavigated ones have a variation of about 10 volts per meter. Or they reduce the uncertainty by a factor of 3. And so is 30 volts per meter really that significant? To answer that, here I show a TMS input-output curve. The way this curve is generated is that the coil is placed on the subject scalp directly above the motor cortex and used to generate a finger movement. A pulse is delivered each time increasing the intensity. And so starting from a very low intensity, I would generate a pulse, and then I would figure out what the MEP or how much the finger moved, and I would plot that into this graph. And then I would move along the x-axis by increasing the intensity, and I would start to get these little dots that are depicted here. And then what you see is that as we increase the intensity, we get a higher and higher MEP or motor evoked response. And at some point it saturates. But more importantly, this change from very little activity being generated by the TMS pulse to high activity generated by the TMS pulse occurs in about a 10 volt per meter range. And so meaning that a variation of 30 volts per meter is quite significant. And so the use of neuronavigated procedures is important. And so there are also other uncertain parameters that affect the electric field. For example, there's coil orientation on the scalp. So the coil might be placed correctly, but there might be errors in how it's oriented relative to the targeted neural site. Finally, there is brain size, which can vary about 12% in between individuals. There's also conductivity, which is also highly variable, not only between tissues, but also within tissues. Here, we only considered uncertainty in the bulk conductivity of each tissue. And when we look at our results for the same ROI, it turns out that coil position uncertainty is the most important one, followed by brain size, which neuronavigated procedures can also account for because they have the MRI. Coil orientation and tissue conductivity affect much less. Now, it turns out that here we're considering the magnitude of the electric field. If you were considering the electric field along a given direction, coil orientation would be important. So that's something to keep in mind. And so it turns out that we just hit the tips of the iceberg. There's a certain level of uncertainty in all simulation results. But however, this is the solver's viewpoint. You give it an input scenario, and it gives you a prediction, while neglecting that reality is really messy. You rarely see an experimental result without some kind of error bars or some kind of estimate of uncertainty. Whereas with simulations, oftentimes you just get a single deterministic output. And so to increase the realism of our simulations, we must take into account all of these inherent contingencies. And my brain research project addresses this need. And so here's what we're working towards. The goal is to make variation-aware solvers that take into account all of these contingencies and produce not only predictions, but also confidence intervals. So what are the sources of inherent uncertainties or possible errors? So there are observational errors. So for example, imaging data has movement artifacts or partial volume effects, meaning that multiple tissues can reside on a single voxel. And so as a result, the output images are corrupted, a corrupted representation of the subject. Furthermore, the position where the coil is placed on a simulation might not be reproducible exactly in an experiment because of limited precision in coil placement. So we have limited knowledge of where the coil is placed. Then there are modeling error. The imaging data is typically segmented into tissue types, so there might be misclassification errors which result in erroneous head models. Furthermore, how the coil is modeled affects the solution. We have done these studies and it turns out that this is negligible, but nevertheless, formally, there is a source of modeling error there. Finally, there are discretization errors. When we solve these things on a computer, our solutions depend on the density of the message used to discretize the head and the order of our numerical methods. Here, I show a figure illustrating how different meshes can result in widely different solutions for the same scenario. In the end, our goal is to create a solver that not only generates predictions for the TMS-induced electric fields, and we also want to predict uncertainty resulting from all of these things that I just mentioned. And then once we have all this, we want to use it to come up with coil placements that not only maximize electric fields at targeted cortical size, but also minimize uncertainty or inherent uncertainty in our procedures. I'll briefly talk about ongoing work I'm doing to get to this vision that I have, to give you an idea of how we're pushing towards this. And so first goal was to remove discretization error. So what we did is we put together all of the methods used in the literature and some new ones and compared them in terms of error to figure out the optimal strategy for reducing error while keeping computational time tractable. If you're interested in using simulators in your experiments, I highly recommend our paper in brain stimulation. It provides essential background for effective use of simulators in an accessible manner. And all of these codes are available and work online, and they work in Linux, Windows, Mac, using MATLAB interfaces. And so another thing we've been looking at is how observational errors affect the fidelity of our simulation outputs. So as I said earlier, MRI images have movement artifact and partial volume effects and other sources of error. Furthermore, to generate a head model using these MRIs, they're typically passed through these segmentation algorithms, and these segmentation algorithms can misclassify individual voxels, resulting in erroneous head models. So to estimate these possible errors, how these possible errors result in uncertainty in the predicted electric field, we treat tissue boundaries as random variables. Here I show a CSF, and different realizations of the CSF differ slightly to account for tissue boundary uncertainty. Then we can run a Monte Carlo to determine the electric field uncertainty due to possible errors in the CSF discretization. And here is an output of the expected electric field and a standard deviation. We are currently extending this framework for tissue interfaces like gray matter and white matter. And so there's not only single subject variability, but there's also inter-subject variability. And analyzing this in principle is quite easy. To first get a number of subject head models, and then identify ROIs in the subject using any functional MRI toolbox. Then for each, we can determine the coil position to E-field maps on Atlas Scout. Here it's plotted on a hemispherical surface. So this hemispherical surface is a transformation from the scalp to a spherical, and the EEG coordinates are used to generate this mapping. And so the color of of these individual hemispheres represents what electric field would be generated if the coil is positioned at that specific position on the scalp on the targeted neural site. And so then you can combine these E-field maps to and integrate them to generate E-field population level information on the E-fields induced if a normalized coordinate system is used for TMS. And so this is a particularly useful example for a researcher studying some kind of disease and wants to come up with a coil placement that will maximize efficacy of their paradigm while minimizing uncertainty across individuals. They can use this to determine a position in terms of EEG coordinates to place the coil that can be used across individuals. And this currently all can be done. It's relatively straightforward. However, these analyses are quite computationally intensive. And so what we're working is in developing fast methods to make doing these analyses on a laptop possible so that this can be used in a practical setting to inform coil positioning. And so this slide summarizes my research accomplishments, and I'll read some of them to you. So I developed a first comprehensive UQ framework for TMS, obtain new insights into factors that affect TMS variability, yes, coil position, yes, brain size, no conductivity and no orientation. And the idea here is to extend these frameworks to develop TMS paradigms that are robust to coil placement errors. And so this sums it up. Thank you very much.